Hello everybody, I'm Steve.
I'm Dennis. Welcome to Natural Growing Through Biology podcast series.
Yay! All right, so we're gonna have some fun, as I feel like we normally do. We're gonna focus on microbes.
That is so weird.
It's new. I know. We're going a different direction this time. We're gonna talk about microbes and microbe functions. Oh wait, no, that's all day, every day. We're going to look a little bit at carbon, residue breakdown, nutrition release associated with that carbon breakdown, because we got some pretty cool data. We were given permission to share some data that we wanted to make sure people had an understanding of, because we're looking into a little bit different environment, a more conventional environment that's starting to incorporate a more biological-minded approach, because nature has solutions. We are just, what Tineo is doing, we're just trying to gather those solutions that nature already provides and make it easy for a grower to use. Whether we're talking about, in this case, cellulose digestion and breakdown, residue management breakdown, or if we're talking about like a compost tea situation, which I think we'll talk about here in the future, where this idea of all of these things nature is doing is fantastic. And what we're trying to do is take some of that, concentrate it doan, and put it into an easy to use format.
Well, yeah, and this is a little bit different than what we normally talk about, because this isn't our normal seed treat application in furrow at the time of This is a fall program that was specifically built for a specific problem, and that's what we want to really talk about, solution.
Biological solutions. So crazy. So we're going to look at some digestion data. We're going to talk about living systems and how carbon is associated with that, and how this composting of material breaks down, how composting functions somewhat, and then releases the nutrients and builds soil. All that good. The cycle of carbon, the carbon cycle, the cycle of life. And last time I was talking about how carbon is the currency of life, and that's a lot of what's going on. Is these microbes that are in the soil looking for food. They're scavengers. They're out there looking for a food source. So when we get into an environment that has a lot of food, they're going to go after it and do their job. So wanted to give a big, giant thank you to Twin State for allowing us to share this data. And that's where this data was collected from. They've been doing, oh boy, how many years? Years of research with us, looking at different products, different approaches, and how to utilize some of these living biological systems in a pretty conventional scenario.
And, you know, when we start to talk about regenerative agriculture and conventional systems, and we can go back, which is the right way, which is the wrong way, but it's utilizing these tools in every different system to achieve a benefit. And it's amazing how sometimes we say, well, you can't do this based off of an application. This is not how microbes work. But we're finding just basically how adaptable they are to these different environments, and how we can utilize these tools in much broader spectrum than we probably originally thought. And that's what's really exciting about this trial, because this is definitely 100% a conventional system, which is now realizing that these tools actually can function and help them and move them forward. And that's what's really exciting about all this to me.
So we're looking at corn, we're looking at Illinois, and there's a little bit of corn and soy grown in those. Just a little bit?
Just a little bit.
A few million acres? Yeah. Okay. So this is an area that a lot of growers are utilizing these crops. They're utilizing similar inputs. They're utilizing similar methodologies. What's the problem that they were having here?
It was, obviously looking at digestion of field debris. Sometimes we have this conversation all the time with growers, where we see whether it's GMO or non-GMO. The GMO obviously is a little tougher.
We see a slower breakdown.
Slower breakdown. It's a little tougher. So then you have in your rotation, whether it's corn on corn or corn following soy or going into soybeans, that you have a residue management problem. And that's what this was originally designed for, was residue breakdown.
What problems occur when you have that much residue buildup?
Well, you just seeding. Basically, drilling your seed is a tough problem. You have too much debris. You don't get good soil contact. Then you have germination issues. You have stand issues. All of those types of things. And so, in order to get those seed drills through the field and get good seeding and good soil contact with these seeds, so you get good germination and all of those types of things, is how do we manage that debris?
Got to build that seed bed.
Exactly. And a lot of times in these environments, we may be taking that crop off very late in the season, which brings in a whole nother issue based off of timing and microbe application and all of these types of things. So, I mean, a little bit here, this is just for reference. I mean, they shared this information with us as, you know, basically an organic matter, 3.5%.
That's nice.
Yeah, pH 6.4.
That's great.
CEC 17.6.
Good
so
stuff.
A pretty good soil environment.
These aren't marginal. These aren't marginal soils.
Correct. So good yields, good soil health, all of these types of things. But again, they had a very specific problem of too much residue, and how do we manage that residue, basically, to grow our next crop.
A little bit of armor is good, but if you keep layering armor on, I mean, thinking about this as a, you know, I'm a knight out there. It's too heavy. It's too much weight. It's too much slowdown. Our systems no longer function the way that they're supposed to. And that's kind of what they're seeing. If you go out in the field and you can see the last 3 or 4 years crop residue out there, which we see pretty regularly, we know we've got a problem. We've got an issue with building that seed bed, but we've also got an issue with nutrient release, nutrient management, which starts to cause decline. We also start to see excessive moisture buildup in some of these situations. And the problem with that level is there are a lot of saprophytes that want to function in that environment. Saprophytes are a good thing. They're breaking down material. The bad side of that is there's a lot of disease organisms that are saprophytes. So we start creating an environment, and we say environment dictates expression all the time. We are creating an environment that is less conducive to beneficial microbe life and more conducive to detrimental.
Yeah, well, and a good example of that is, you know, I love to pheasant hunt. And going through Kansas or Nebraska, I know, Kansas or Nebraska or Illinois or wherever I have basically wherever I have gone and done these different
wherever your
activities,
boots take you.
Exactly. The guys I hunt with sometimes get frustrated because I'll be walking through a field and I'll be laughing whether it's a tillage operation or a no-till operation, where I can tell them the crop rotation just based off the debris that is not broken down within the soil environment. Sometimes they're like, just shut up. We don't want to hear anymore.
Stop building the watch. We just want to know time.
So, you know, sometimes when you see that, it's like you say, Steve, you tie up a nitrogen leg in the springtime because that is being utilized to soil digestion, all of these types of things, let alone what we just talked about. The specific function of this trial was residue breakdown for better seeding within that soil environment. So, you know, to kind of move on and look at this a little bit, basically the crop management was corn on corn. So excessive debris was the big thing. You know, fall treatments were made on 11/8 of 19. So this has been going on since basically 2019. They've been doing these trials. So it's replicated trials over a long period of time, which is pretty exciting to see, because a lot of times a one-year trial doesn't give us all the information. We like to see those multiple years so we see the consistency in the function of a product. And that's what's really exciting for us, is because that's a lot of times what we talk about. When we build microbial inoculums, we build them for consistency. Being able to see the same results time and time and time again. Because that's very important as a grower when you start to make these decisions about what products to use and how to utilize them within an agriculture environment.
Yeah, if you've got a tractor out there that you can't rely on, you're not going to be excited to use that tractor. We're trying to build and test, don't guess. We're doing testing of our microbes every single batch. So we're trying to make sure we maintain consistency so we have year after year after year performance.
Yeah, and so this was a field chisel plowed 1 week after application. Field cultivated in the spring. Crop residue photos were taken, which is really kind of cool.
I know.
Because, I mean, this is how we measure residue breakdown. And then some of the results that we'll talk about that we saw, which was in addition to that. Spring treatments applications were also made on 5/4 of 2020. And planting date was 5/4 of 2020, and 100 pounds of nitrogen was broadcast as UAN following the planting.
180,
Yeah, so
pretty normal.
Yeah, so pretty much all data that they shared with us is this is the corn program that they're utilizing in this area.
Yeah, and they even shared some of the software they're using and how they're doing the actual evaluation, which is pretty cool. And I've been playing with Canapeo, I think that's how you say it, and there are a bunch of different apps that basically you can use your phone, you can use your camera, and you're taking a similar size photo of a similar size area, say it's a meter by a meter, or 2 foot by 2 foot. You got to do the same size area, whether you're doing a control or you're treated. But you take the picture, and then the software is analyzing for specific colors. In some cases, we can utilize it for green colors. In our cameras and our phones, and that software is fantastic at identifying differences in colors. So it can assign a different number of pixels associated with each of those colors and then give you a pretty exact percentage. So in this case, they're looking at field debris. You've got your dark brown soil, and then the camera and the software is able to pick up all of that stubble and evaluate all of that stubble and look at how much is there and left.
Yeah, so the accumulation of residue and then nutrient tie-up or release, obviously, was some of the things. Drilling, planting issues, as we talked about.
That's the big one.
Yeah, bed prep, and then utilizing the software, or basically the things, that come into what we were looking at here based off of this trial. And the exciting part about this is this was just not one product.
No.
This was just not our biodigester.
Yeah.
There were multiple different biological products used from the market out there within this this given trial. They were looking,
8 other products.
They were looking, So, when you start to talk about this was a lot of work for Twin State in order to do, start to really find out and what was work. So the treatments were applied in the fall. Fertilizer broadcasted at 86 gallons per acre with 23 nitrogen, 50 phosphorus, 75 potassium, 15 sulfur, and then a little bit of zinc and a little bit of boron. This was all added as a package with this biological within the soil environment. And they were applied the same thing again in the spring, which we talked about the planting dates or the application dates that we had talked about earlier.
To encourage breakdown, because we know that some of those nutrients are required for breakdown. Nitrogen is critical for breakdown of this residue, this material, as is potassium, as is phosphorus. We need that energy. So a lot of these nutrients are required to help speed up and boost this breakdown process.
Yeah, the digestion process. And as we look at that, I always refer to this as pre-digestion. Basically, we are breaking this stuff down through microbial activity, which is then basically building that into a soil aggregate, which is keeping that stable within that soil environment. So next spring when we plant our crop, that nutrient is there waiting for that root, waiting for that rhizophagy cycle for those microbes to start that digestion and make it available. It's pre-digested. It's there waiting for them to utilize it.
It's a relay race. We're handing off that nutrition right to the plant. Here's some photographs, which are difficult for people that are listening to see. But what they did is they're out there mixing product up and they are out spraying. So if we're talking about 9 different products with a minimum of 6 replications, that is a lot of work. They did a lot of, so again, we're big thank you to Twin State, a big thank you for sharing the data, because it's really cool what they were doing and how they were able to do it. So they go out and they spray each of these plots like we were talking about, and then they're utilizing the software for evaluation and looking at the photos before and after to evaluate the percentage of breakdown of the material. And that's what this data is looking at. So looking at the check, surface residue percentage was 33.4%, with the biodigester at 30.6%. So right around a 3% breakdown. That doesn't sound like much. That is one of the best method, the best tools, the best products, the best performance that they saw in residue breakdown. A lot of the other products, 31.8, 31.7, 31.6, 32.5. So that 30.6, that 3% breakdown, is actually a pretty considerable amount of breakdown. It's 9.15% better breakdown, almost 10% better breakdown of that surface residue than we saw from the check.
And one of the things that we see in this residue breakdown is not only the breakdown at that 10% that you're talking about, but when you start to talk about that material of those seeders, basically those drills going through, and rather than that debris basically being resilient within that soil environment, and basically you're laying the seed on top, it can cut through.
Yeah.
It's almost like it started to decompose. It's not completely broken down, but if you grab it, it just breaks apart.
The picture and the software, it does a good job evaluating total percentage, but there are other fine details. It's like it's using your senses out in the field, which we talk to people about all. going out, digging in, it's smelling the soil, it's touching the soil, it's feeling the soil. Feeling this material, when we go to these sites that are a control versus a treated, oh, absolutely. The material has a completely different feel. Even though visually the structure looks similar, when you pick that material up, it's spongy. It falls right apart. And as our chisel plows, as our equipment is going through, it falls apart and breaks down even more, which is exactly what we want to happen.
But not only did we see a 10% basically increase in residue breakdown, we also saw a yield bump. And that was the kind of the thing that they looked at, that this was something that they weren't even looking at. This was just specifically make it easier to plant. But we saw the check was at 208 bushel per acre, and where the biodigester was used was 218 bushel per acre.
10 bushel? That's no slouch.
Yeah, I mean, basically a 5% yield increase is kind of what we were looking at based off of utilizing the system, and that's kind of what we talk about, that pre-digestion. And we'll get into that a little bit later in the presentation of why all this happens. But so the cost basically was $9.04 an acre. So 2 applications, it came out to $18.08. But the great thing about this is their ROI was $22 per acre by utilizing the biodigester within that field environment. Based off a yield bump and cost, they saw a $22 per acre yield increase, which is very exciting, especially, I mean, they were talking $4 corn here. So as we start to look at how do we manage a better ROI based off our investment when crop prices are extremely low, and we're looking at that going into this next season with everything that's going on, is how do we make more money? And that was the thing that we were so excited about. I mean, as you referred to this, Steve, this is gravy.
That's great. So the focus of this is breaking it down, building that seed bed so that we have better plant stand, better plant germination, better plant emergence, better plant growth. And then what we see from the biological side is, we do all that, but then we're also releasing the nutrition. We're utilizing plant growth promoting rhizobacteria that help that plant establish, grow that plant. So all of this yield, all this bump, that's the delicious stuff. That's the gravy. We get it.
Yeah, you can never have too much gravy.
That's right. It's like Thanksgiving every day.
That's exactly, or a chicken fried steak. Yeah, so just a little more information they gave us on the breakdown products of us and the competitors. This was a little bit different, but we showed the check was 195 bushel per acre. This was the broadcasted out.
In no-till.
In no-till. We saw 211 basically with the biodigester. So roughly about the same. What is that? What's
8.2% increase from 195 to 211. That's a considerable bump. I'm pretty happy. And then comparing to competitor A and competitor B, they were at 205 and 203. So our 211 bushel did pretty darn well. They're pretty happy with that.
Yeah, and we obviously saw in the broadcast scenario where we're doing the entire field, broadcasting it, we saw a better yield bump than when we were just doing a dribble band basically within the field environment. You know, on that we only saw 207, but still a bump.
201 versus 207. 201 the check, 207 in the treated. But that does kind of make sense, as we're broadcast isn't our favorite, but remember, they were pretty immediately after application chiseling that in, working that in, incorporating that into the soil. Light incorporation we talk about regularly it's so important, so critical. A lot of that is maintaining moisture levels so that our microbes don't dry out. If that material is up on the surface, we've got wind, we've got evaporation, we've got loss, we've got sunlight, we've got UV, we've got all these different issues that are damaging the microbes, slowing them down. A light incorporation and a little bit of soil on top of that helps protect it. As long as we have good gas exchange, now those microbes have better access to a lot of the factors that we talk about are critical for life. That water, that air, that light, all of that stuff is in a better environment. So it makes sense that we saw that.
You know, and talking about basically, you know, I always say this, at Tanya, we figured out a long time ago how to put these guys into assisted dormant state so that you can wake them up and put them out into that soil environment. And this is one of the scenarios that we talk about, is basically keeping them alive, basically incorporating them a little bit in that soil environment. But you know, another thing it reminds me, as we talked about when we were talking to Twin States and we were building this system, is we kept stressing how important it was to buffer those microbes with carbon as we tank mixed them with that fertilizer and all of those things that we had talked about before. And they actually did a trial where they didn't use carbon and they did use carbon, and they saw a significant bump by adding that carbon component to buffer those organisms and protect them as we put them into that tank mix. And to me, that's really exciting, because as we start to look at this in these conventional systems, how we can utilize these microbes by buffering them with that carbon source to put them in, you know, because we always say, you know, the high salt index, all of these tank mix based off of environment is not great for those microbes. But if we buffer them and we get them out right away, they're living and still able to do that function within that soil environment, especially like you said, when we start to do that incorporation. You know, we always talk about, if I'm in the springtime and I'm applying a biological inoculum, I want it around that seed. So at that point of germination, we talk about with rhizophagy and sharing those sugars or those communication molecules and all that, we want it right there. But in this scenario, this is a little bit different, where we're trying to get digestion across an entire field. So a field application or broadcast application in this environment makes sense.
Oh, absolutely. So looking at some of this even deeper, they did more trials, they did more research, and in this case they were looking at 3 different locations in Illinois, and again, replicated study, looking at, in this case we're looking at OP8 plus carbon compared to a competitor product, and in this case we saw about a 4 bushel yield increase over control and a 7 bushel increase over the competitor. It's a pretty name brand. Yeah, pretty significant. We also saw the test weight. The control was 56.35. The competitor was 56.82. The OP8 ended up with a 57.58 test weight. So we had better yields and we had better test weight. That's more gravy.
That was more gravy. And you know, we start to talk about that. So why do we see all these things? And it goes back to what we talk about. This is how nature does it. When we start to look at basically that breakdown, that the microbes basically building an environment, I refer to it as habitat, a home, basically for them to live in. And some of the benefits that we start to see from that is we start to see soil structure. We start to see they create a relationship with the plant. And we look at the trap of nutrient or nutrient availability. That's the big thing. Those are the things that we start seeing in this conventional system, is that release of that nutrition within that soil environment. And you know, you always say carbon is king. I mean, we see that carbon source being developed within that soil environment through that digestion process. And then again, the signaling and the communication. These are all the things that are happening within that environment, as what we see is why this all makes sense.
Yeah, you were just talking about the importance of utilizing some of that humate, some of that carbon in a tank mix situation. It's buffering, it's capturing, it's holding on to a lot of that. So this organic matter, this residue that's on the surface of the fields, that's a big part of what it's doing. And it functions like a sponge. That is a big part of what this organic matter's job is. We're holding on to more moisture. Depending on that structure, we're allowing better gas exchange. We're allowing these things that we need to happen for this to become less of just dirt and become a living skin. This rhizosheath, this rhizosphere, this O horizon, this A horizon. We want more life in this environment, and a big part of building that life is creating that habitat. It's creating those homes.
It is, because you know, Bruce used to say basically animals, plant, people, they all need basically 5 things: light, heat, air, nutrient, and water. And if we start to think about that, that's what the microbes need. That is also what the plant needs. But only when these are unbalanced can we have optimal nutrition. We talk about the law of minimum, and we so often concentrate on a deficiency or an excess being our greatest limiting factor. But as we know, a lot of times our soil environment, or our lack of diversity of that microbial community, or the breakdown of that organic matter and that nutrient cycling, sometimes creating that habitat, is basically creating that living skin that we talked about. Because really, if you think about it, microbes are basically just water, air, nutrients, and carbon. That's what they are.
And that's what they're utilizing to. That is the basic formula of life. These microbes, these bacteria, these fungi, the nematodes, protozoa, all of them, humans, plants, we're all basically the same recipe. We're doing the same jobs. We just have different slight functions. Plants have a primary job. Their primary job is to take carbon from the atmosphere and make it into a usable form. CO2 isn't utilizable as its molecule by most organisms. Very few organisms have the capacity to fix carbon into a utilizable form. That's what plants do. That's what all of that structure, that's what all of that material is. It's taking that carbon out of the air and creating structure out of it. Microbes, most of them, purple non-sulfur, and there's some other photosynthetics that are down there, they have the capacity to do that. Not necessarily as well as a plant, but they can do it. But these microbes want to take that carbon and they want to create what's known as a microbial carbon pump. And we need to focus on how important that is. We've been talking a lot about residue management, how important it is, how we're building soil organic matter, how we're building soil carbon. So we focus a lot on how important this plant material is, but we need to focus the other side of that, and research is showing more and more and more how critical these microbes are. So if I go to an environment that has been down the aisle of death a whole bunch of times, we're using a lot of tillage, we're doing a lot of things to degrade the biological side of these soils. Even if we have decent residue, we don't convert that and we're not building that organic matter, because research is showing that 50% of the soil organic carbon is dead microbial biomass, or also known as necromass. Some research that we got from Nature Microbiology, we showed that microbial necromass can make up more than half of the soil organic carbon. Hence, we suggest that next generation field management requires promoting microbial biomass formation and necromass preservation. You mentioned this idea of erosion, of loss, of stability, of these microbes creating homes and creating these microaggregates that are then created into macroaggregates. That is all stabilizing the soil environment, because they want a house. They want a nice, healthy, happy, safe home for them to raise their little baby microbes in. Well, that's life.
And that's really what Twin State was doing in this study, is exactly, they are utilizing these organisms basically to build soil health and build soil function. And that function, again, the idea of this was to break down that debris to make it easier to seed, but all the benefits, that's sometimes what we don't look at based off of agriculture and basically regenerative agriculture and building soil health, are all of these other benefits that we get to see based off of overall plant health, soil structure, water holding capability, carbon, nutrient availability. I mean, the list just goes on and on and on.
And it's so exciting looking at these homes, looking at these structures, because we talked a lot in the past about this idea of microbes binding soil particles. So I want to take a few minutes and kind of dig into that a little bit more and refer to it as EPS before, usually extracellular polysaccharides. It's kind of shifting. The verbiage is still EPS, but now we're starting to call it extracellular polymeric substances, and that's because research is showing more and more and more these microbes are not just using the sugar as a glue to bind things together. They are putting all sorts of stuff in there. It's not only sugar, they're also pumping in proteins, so we've got a little bit of nitrogen storage in there that the plant and the microbe can utilize. We also see a lot of enzyme deposition in there. So if we look at a soil, a mineral, an actual physical particle, we talk about, the chemolithotrophs, how microbes, the bacteria, are the first ones starting to break this material down. So when you look at those initial organisms that are breaking down that rock, the microbes are creating this biofilm, and they're dumping massive quantities of enzymes into it. So when we start talking about, oh, my soil has a lot of calcium, but I can't get the calcium. My soil has a lot of phosphorus, but I can't get that phosphorus. These microbes are creating a structure around those particles and then flooding it with these enzymes. So those enzymes can break this process down. It's putting an environment around the microbe, around that soil particle, around that organic matter, so that it can kind of naturally break down. Because that's the beauty of enzymes. They can function over and over and over again. So if a microbe surrounds a small clump of cellulose or fiber with this biofilm and binds it together, and it floods it with cellulase, The microbe kind of sits back and waits for a lot of the sugar to be broken off and spit off and broken down so they can access it. The same thing is happening with our physical minerals and our structures around those minerals. The microbes are creating the zone around it, allowing the enzymes to break this off. Not only that, but this biofilm, being a high carbon material, is also fantastic at trapping the nutrients. So when we get into the idea of this rhizosheath, these sticky root structures that is full of all this microaggregate, that is all bound together by these EPSs, and these EPSs are holding on to enzymes. They're holding on to a lot of defensive molecules as well. These microbes can sense what is going on in the environment. We've talked a little bit before about this idea of chemotaxis, this idea of microbes using different communication molecules. They sense what's going on, and a lot of those molecules get trapped in this EPS, allowing the microbes to sense more of what's going on in the environment around them. There is, sorry, I'm getting kind of worked up and excited because it is so cool, and we see significant gradient differences in this EPS zone. We often talk about bulk soil versus the rhizosheath. This is a big part of why. If a microbe is pumping out hydrogen in an environment to lower the pH and it's in just a wet bulk soil, it can get flushed and flooded away. Inside of this polymeric zone, it is trapped, it is stabilized, it is holding on to all of that, which is just, it's so exciting. We also see massive levels of DNA exchange. Microbes are constantly spitting out DNA molecules. They're kind of constantly talking to each other. They even have specific structures that they'll utilize called plasmids. So sometimes the microbes will package up a little functional packet of DNA to do a specific job called a plasmid. They can spit it out. They can dump it into this environment where it's stable. It's protected from sunlight. It's protected from drying out because the EPS holds on to more moisture. And the microbes can then exchange this DNA in a safe environment. So if there's a lot of phosphorus and we have a few organisms that are better at it and they can produce a plasmid that has more phosphatase enzyme production capacity, other microbes can start utilizing it so we can start to have more phosphate solubilization, more calcium availability in this sink, in this spongy structure to hold on to it, to allow that plant to access it.
Well, so really what you just said, Steve, I'm going to give the Reader's Digest version. EPSs are fundamental for microbe life and provide an ideal environment for chemical reactions, kind of what you were just talking about, nutrient entrapment, holding on to that nutrients, and protection against environmental stresses. So, I mean, when we start to talk about salinity, we start to talk about drought, we start to talk about pathogens, all of these, this is what they're doing, basically, within that environment. That's really all what you just explained of how that entire process happens. I mean, that's what we talk about all the time, and that's a lot of what we just saw in this trial that was done. They're doing their function based off of residue breakdown.
It is an insulation layer. The thicker the microbial biofilm that is being produced, the more insulation. The beauty also is the microbes are dumping garbage into this insulation. We see examples where you mentioned sodium. They're also dumping heavy metals into there. They're dumping chemicals and molecules in there that they don't want the plant or themselves to be exposed to. They are using this purposefully as a dump ground to protect themselves. This is a fantastic insulative layer that they're utilizing and they're building constantly.
Yeah. And so, I mean, let's talk a little bit about, I guess, from the idea of how do we apply these guys.
Let's make it work.
Yeah, so, you know, basically we call it the fall program, the fall soil primer program. Basically trying to accomplish things post-harvest breakdown. You know, you talked about light incorporation, if we can have that. A lot of times we look at this is for speed up a digestion, to break down that environment, again, putting those microbes in an environment where they actually
can do their job.
You know, sometimes I look at that when we start to implement these practices of utilizing, for an example, biodigester in the fall. Depending on when the corn comes off, basically, you know, I mean, obviously, if it's late winter and the ground is frozen, this probably doesn't make a lot of sense.
Microbes need liquid water.
Exactly. And so we need to be able to do that. But if we have a relative short period of time, let's say late fall, ground isn't frozen yet, we can still apply these guys. We can have them in that environment because they will continue to work and do that digestion over the winter time.
Yes.
Just like you think like a compost pile.
It gets
Compost pile gets hot, it heats up. These microbes can do that exact same thing within that soil environment.
Every time they break one of those cellulosic bonds, the beta-linked cellulose with the cellulase, every time they break one of those bonds, a small amount of energy is released. Bruce used to talk about that. These enzymes are basically a magnifying glass, focusing energy to break that bond, and in doing so, energy is released, and that energy is part of what keeps them up and running and functioning.
Yeah. So, late fall application, we're going to see better digestion. But if we can't do it, a lot of times of doing that in the springtime, prior to planting, when we can get out into that field to help with that residue breakdown, we may not see the same level of breakdown that we saw in the trial, because it does take time. It takes time. But think about that, all of this nutrient, everything we've talked about today, based off the breakdown, the nutrient release, the soil, all of that, carrying that crop through. A lot of times when we get into that late summer time, where we're running out of nutrition and that plant isn't functioning and those roots aren't functioning at the highest level. Now, all of what we've done, the microbes have done from that spring application, now is available to that plant in that late season. I see that all the time, carrying that crop, helping it finish, basically to get across that finish line. It's just ran a marathon. It's running out of gas, and that's just, what do they call those little go-go's that you can drink, or proteins that you take basically when you're doing a marathon? My daughter would know. She does them all the time. But it gives you that energy and that food that you need to finish the job. Yeah. That's a lot of times what we look at. And sometimes we look at which product to use. We talk about the biodigester here in this study being applied to the no-till situations. We had the biodigester being applied as residue breakdown. I've had some growers that, as you talked about armor on the soil, we want to maintain some of that. Yeah. I had some growers that were utilizing biodigester this was in Kansas, but they started to look at the idea that we were breaking down our residue too fast. And so they went to Spectrum, and they were using Spectrum as the fall primer program. There's one grower in Kansas that told me last year, it was interesting. He's been doing the fall primer program for a long time, and he went 1 year because he wanted to save money. He didn't want that expense going into the fall.
How did it work out?
He said he would never do that again. Yeah, he saw a decrease in yield and ROI by doing that. So all of these things, how do these tools fit in? How do we get them out there? We talked about adding carbon in the tank mix.
Critical.
But don't forget about the food, the fish, the humates, the kelps, the molasses, adding that carbon to help these microbes basically survive and do their function as designed. And with that, I guess we'd like to say thank you, everybody. The microbes, as we always say, can only do their job if they're given the chance. And
you miss school, you miss out. If the microbes aren't there, the plant's missing out.
Exactly. So, in this situation, like you say, we have a lot of ways to utilize microbial communities to enhance agricultural processes.
It's a tool. It's one more tool.
It's one more tool, and it doesn't matter whether, as we just said today, we are 100% organic or organic minded or conventional. As we all have to start somewhere, going down this path of what I call biological farming. And with that, we'd like to thank everybody for joining us, Natural Growing Through Biology podcast. I'm Dennis.
I'm Steve.
And look forward to seeing you here again soon.
Thanks, everybody.